Okay. Hello, everyone. Thank you for joining us in this session of Convey Seminar Series. It's a pleasure to virtually welcome Professor Antonietta Mira, an accomplished Italian computational statistician with remarkable contributions to the field of Bayesian statistics. Professor Mira is currently a professor of statistics at both the University of Lugano in Switzerland and the University of Insubria in Italy. She also holds two PhDs, one from the University of Toronto in Italy and the other one from University of Minnesota in the United States. And her research focuses on the development and application of Markov chain, Monte Carlo methods for Bayesian inference. Among her numerous honors, Professor Mira is a fellow of both International Society of Bayesian Analysis as well as the Institute of Mathematical Statistics. And beyond academia, she is passionate about promoting statistical thinking to the public, and he has actively involved in exhibitions, books, and media outreach. We are delighted to have her with us today and look forward to her talk. Professor, thank you for accepting our invitation, and over to you.
Well, thank you for inviting me, and thanks for Fabrizio Ruggeri for suggesting my name. It's a real pleasure to be able to tell you a little bit about my research. And specifically today, I will talk about the data intrinsic dimension. This is a research line that I've been working on for a while with a few colleagues. It looks like a football team. On one side, we have the colleagues from SISSA in Trieste, Italy. They are all physicists. On the bottom, there are the statisticians. Some of them are from Università della Svizzera Italiana. Some of them are from Queensland University of Technology. By the time they were working on this research project, most of the collaborators were PhDs. Plus, there are two other principal investigators, Professor Alessandro Laio, who is professor at SISSA, and Professor Kerry Mengersen. So this is a real interdisciplinary project ranging from statistics to physics. You have seen my colleagues from physics, SISSA. They are almost the majority of the team, of the research team. And some domain experts have also joined this effort that started in 2019, and it's still ongoing. And this research line led to 5 methodological papers, 2 more applied papers, and 2 proceedings. So let me ask a question and start with an example. So you see data here that have been collected in 3 dimensions. And here you see 2 different perspectives of the same data set. And the question is, can you guess the dimension of the support of the data generating process? Meaning, is this data really free to cover the 3-dimensional space, or is it constrained to live on a smaller dimensional manifold? Well, looking at the left-hand side perspective, you see that maybe there is something going on here. And if you rotate the data, you realize that this data set is constrained to live on a 2-dimensional space, 2-dimensional manifold, even if the coordinates are 3D. So this is a Swiss roll motivating example where capital D is 3. So for each data point, we have 3 coordinates, while lowercase d is 2. And lowercase d is called the intrinsic dimension of the data. And it's really relevant to identify this intrinsic dimension. For example, if you want to perform dimensionality reduction, if capital D is very, very large, and you want to project your data on a smaller dimensional manifold, you need to decide what is the dimension of the smaller manifold where you want to project your data without losing too much information. Estimating this lowercase d is also important for feature selection or for unsupervised or even supervised learning. In a sense, this intrinsic dimension, lowercase d, can be interpreted as the lower bound of the number of variables that are really needed to describe a system. So we eliminate redundant variables and we only keep those variables or those combination of the original variables that carry the relevant information. Okay. So in order to estimate this intrinsic dimension, we try to answer some open questions. As I said, often a subset of the original variables, capital D, or nonlinear combination of those variables are sufficient to describe a real-world data set. And the question is how many and which one of those variables or which nonlinear combination is sufficient to summarize the data set without loss of information. This is the real question that we started off addressing, but there is a related question, which is the question of the appropriate scale at which we should analyze the data. And these two questions are often considered as unrelated, but they are indeed strongly entangled. And we address the first question and the second one together, estimating this intrinsic dimension. And to do this, we introduce a unified approach which self-consistently estimates the number of relevant dimensions in the data set and the scale at which we should look at the data, bypassing the noise and really allowing us to see what's going on. What are the applications that we have analyzed using this methodology? Well, they're very, very different. They go from protein folding to fMRI data, gene expression, some financial risk data, sport data. Of course, we have analyzed the evolution of the COVID-19 pandemic using this methodology. We also studied network, both directed and undirected, weighted and non-weighted. We studied the intrinsic dimension of the output of a Markov chain Monte Carlo simulation. We studied the intrinsic dimension of convolutional neural network and transformers in large language models. So the applications range from, finance, sport, biological, health-related data, all the way to machine learning. And in all these applications, a simple topological feature like the intrinsic dimension allows us to uncover a rich data structure. And I will talk about some of these dimensions. So let me go deeper into a formal definition of this intrinsic dimension. From a statistical point of view, it is the dimension of the support of the data generating distribution. You can also think of this intrinsic dimension as the fractal dimension if you're a mathematician. Indeed, the intrinsic dimension does not need to be an integer. And in the literature, there are very different approaches to estimate the intrinsic dimension. Some are projective, others are topological. We focus on an approach based on the nearest neighbors. The other approaches we don't like because they lack a sound statistical interpretation. So the aim of our research line is to estimate the intrinsic dimension, and we treat this intrinsic dimension as a parameter that we want to estimate. And the problem is that this intrinsic dimension, as I will show you on some example, is scale dependent. And this is why the two questions of estimating the intrinsic dimension and identifying the noise and defining the scale at which we want to look at the data to bypass the noise are very much connected. And we want to answer these questions using some statistical guarantees, because we want to estimate the intrinsic dimension with the corresponding uncertainty of our estimators. And we also want to have some asymptotic results on the asymptotic distribution of our estimator. So let me tell you how the intrinsic dimension is hard to estimate, because it's related to the noise around which the data is collected. So in this case, again, the data is three-dimensional. All my pictures are three-dimensional. You can imagine why. So the data is collected in 3D, but is really constrained to live on a one-dimensional line. But the data is collected with noise. And so if we look at this data set too closely, it seems that the intrinsic dimension of the data is really three-dimensional. And if we zoom out a little, we really see the one-dimensional structure. So the question is, how much do we want to zoom out? So the first problem that we have to solve when estimating the intrinsic dimension is the scale at which we want to look at the data to bypass the noise and really identify the structure in the data. The second problem is that sometimes in a given data set, there's more than one intrinsic dimension. For example, this is a simulated data set of 5,000 points collected in a 9-dimensional setting. So capital D is 9. But really, the first 1,000 points have intrinsic dimension 1, so they are generated from a one-dimensional Gaussian. Then the second 1,000 points, the green one, have intrinsic dimension 2, and then 4, 5, all the way to the last 1,000 points that have really intrinsic dimension 9. So in this plot, you see a three-dimensional projection of this 9-dimensional data set. And you see that it's very hard to identify the intrinsic dimension of this data set, also because, for example, the first 1,000 points are in part totally embedded in the second 1,000 green points. So the black are embedded in the green and the blue. So this is the second problem that we need to identify. The third problem is related to the fact that sometimes data is discrete and sometimes data is continuous. So this is, for example, some data on a paper in JRSSB, where we have remote sensed chlorophyll data in RLC, and the sensors are discretely placed on the C, and so the data is discrete. So all the theory that I will tell you has been first developed for continuous data set and then extended to discrete type of data. So while solving these three problems, we have proposed in various methodological papers different estimators of the intrinsic dimension. And so the first estimator is called the two nearest neighbor estimators. Then we have the GRIDE estimator, the Hidalgo, the Bayesian nonparametric Hidalgo, and then we have adaptive estimators. So I will start with the two nearest neighbor estimators because it is the simplest to understand, and then we'll extend it to the GRIDE estimator. And as we move down in the list of estimators, we will also have to enforce stronger assumptions for those estimators to have good statistical property. And the two nearest neighbor estimator is the one that requires the weaker local homogeneity assumption of the data. The GRIDE estimator, on the other hand, is more robust to noise in the data, but requires stronger local homogeneity assumption. The Hidalgo and the Bayesian nonparametric Hidalgo estimator are robust to setting where there's more than one intrinsic dimension embedded in the data. And then we have the adaptive intrinsic dimension estimator that allow us to identify the correct scale at which we want to look at the data to really see the structure in the data. So the two nearest neighbor estimator works like this. So suppose you have your matrix of data. So you have n data points collected in capital D dimensions. So again, capital D in this case is 3, and each point is, or each statistical unit in your data set is a point in a 3-dimensional space. So let's look at the ith unit in the data set, and this is this point xi. We indicate with xi1 the first nearest neighbor to xi, and distances can be measured with any distance you like. So the methodology that I will introduce is agnostic on the distance that you use to measure distances. You can think of Euclidean distance or something else. And we let Ri1 be the distance between xi and its first nearest neighbor. And similarly, we will have xi2, the second nearest neighbor, and Ri2, the distance between xi and its second nearest neighbor, and so on. Now, the first result that we can prove is that under some local homogeneity assumption of the Poisson process that is counting the number of points in a ball, this is the only assumption that we need, that this Poisson process is locally homogeneous, locally in the scale of the second nearest neighbor. We can prove that the ratio of distances second to first nearest neighbor from any data point has a Pareto distribution, where the parameter of this Pareto distribution is the intrinsic dimension of the data. So this is a very important result. And with this result, we can use the usual statistical theory to estimate this intrinsic dimension. And we can do this estimation both from a maximum likelihood point of view or from a Bayesian point of view. We have developed both methodologies. So this is pretty much the notation that we need. So xi, xi1, first nearest neighbor, xi2, and so on, second and higher order nearest neighbor, and Ri1, Ri2, and so on, the distances from first and second nearest neighbor. So let me show you another image, again, to convey the same idea. So here again, we have data collected in 3 dimensions that are really constrained to live on a 2-dimensional surface. And notice that the surface on which the data is constrained to live does not need to be linearizable, nor linear. So it can have holes or anything. So let me show you an example to put this into a context. So suppose we have 5 subjects, and on each subject we have 3 measurements. Again, capital D is 3: body temperature, heart rate, and blood pressure. So capital N, number of points, is 5, and capital D, number of measurement or information that we collect on each statistical unit, each person, is 3. Now, suppose that the first 2 persons are really dead bodies. Okay, so their body temperature, the heart rate, and the blood pressure, they are all the same. They are dead bodies. So those 3 measurements have no degrees of freedom. Their intrinsic dimension is 0. They're fixed and they cannot move. On the other hand, suppose the third person is a healthy person. And in our body, there are mechanisms that create relationship between our physiological measures. And we can think, for example, that as the heart rate goes up, the blood pressure responds in some way. So there are some relationship between those measurements. And those relationships constrain these 3 measurements to live on a smaller dimensional manifold. So they're not free to vary and to move in this 3-dimensional space. If you have a healthy person, our body puts constraints on those measurements. And suppose person 4 and person 5, for example, are diabetic person or have some disease. And this mechanism that put constraints on these measurements is broken. And so those measurements go everywhere and they fill in the 3-dimensional space. So this is a setting where a subset of the data has intrinsic dimension 0, another subset has intrinsic dimension 2, and yet another subset has intrinsic dimension 3. This is, of course, a made-up example, but gives you an idea that using this intrinsic dimension, we can ultimately also cluster the data and use the intrinsic dimension of the manifold where the data lives as a clustering methodology. So going back to our picture, if you look at person 1, the first dead body, the closest nearest neighbor is most likely the second dead body because their measurements are pretty much the same. And the second, and Ri1, is the distance between 2 persons, which probably is going to be close to 0. And then the second nearest neighbor is maybe one of the non-healthy person. and this is person 4, and our I2 is the distance from person 1 to person 4. So this is when we focus on the first subject in our data set, but we look at nearest neighbor for every subject. For example, if we now focus on person 4, the first nearest neighbor is the other person that has, for example, diabetes, and the second nearest neighbor is the healthy person, and so on, right? So for each statistical unit in the data set, you look at the first and second or higher order nearest neighbors. Alternatively, what you can do is, again, focus on a subject, and instead of looking at the nearest neighbor, we look at a ball of radius given, say a ball A, and we count the number of points within this ball, and then we look at another ball, a larger ball, say ball B, and again, we count the number of points within this ball. So we have Ka, which is the number of points within ball A, Kb, which is the number of points within ball B. And again, using the same assumption that I've given you before, that is this local uniformity of the Poisson process that is counting the number of points within each ball of a given radius, we can prove that the number of points in the smaller ball, given that we know the number of points in the larger ball, so Ka given Kb as a binomial distribution, with Kb as the number of points, and the success probability of this binomial distribution depends on the intrinsic dimension. So again, using this distributional result, we can estimate the intrinsic dimension. So let me give you an idea of the type of theorems that we prove in the methodological papers. So let's go back to the ratio of second to first nearest neighbor, or more generally, we let n1 and n2 be two positive integers with n2 bigger than n1. And we look at the ratio of distances from every point I in the data set of n2 distance. So the distance to the n2 nearest neighbor, for example, can be the 10th nearest neighbor, divided by the distance of the n1 nearest neighbor. For example, it can be the 10th nearest neighbor. So n1 and n2 can be anything. We can prove that the distribution of this ratio of distances as this form, so we can prove analytically, we can find the distribution of this ratio of distances. And this proof was in a PhD thesis of Francesco Denti, who is now assistant professor in Padova. And yes, assuming that the Poisson point process that counts the number of points in balls is locally, as locally uniform intensity and not globally uniform intensity. And by the way, pretty much all papers before ours assume that this Poisson point process has a global uniform intensity. So our research line is the first one that has made this assumption less strong. It's only local homogeneity of this Poisson point process. We can prove this distributional result. And when N2 is 2 and N1 is 1, this distribution becomes the Pareto distribution. So when N1 is 1 and N2 is 2, that distribution is the Pareto distribution. And by the way, that distribution, we looked around and we couldn't find this distribution anywhere. So it's not a known distribution. As far as I know, if you or anybody knows this distribution, please let us know. But not even the referees were aware of this distribution, which is in a sense a generalization of the Pareto distribution. So having this distributional result, we can estimate this intrinsic dimension, which is now a parameter of a distribution, either by maximum likelihood, and here is the maximum likelihood estimator, and we can have the resulting confidence intervals, which has this lower bound and this upper bound, using the quantile of the inverse gamma distribution, because this estimator has an inverse gamma distribution. Or we can take a Bayesian approach. So if you're a Bayesian, you put a prior on all the unknown parameters. So in this case, we have only one unknown parameters, and which is the intrinsic dimension, which is the quantity that we are trying to estimate. And we put a gamma prior on this unknown parameter. Why a gamma? Because this is the conjugate prior to the Pareto distribution. So we are treating the 2 to first nearest neighbor distances. So with this gamma prior, the posterior, so the distribution of D given the data, and here the data is really those mu's, those ratio of distances, is again gamma, and we can work out analytically how the parameters of the prior are updated into the posterior, and we can use the posterior mean as our estimator. So this is the two nearest neighbor estimator, both in the frequentist and in a Bayesian setting. The GRIDE estimator, which generalizes this two nearest neighbor, relies not on the Pareto distribution, but on this distributional result. And typically, we take N1 to be N2 divided by 2. But this is one of the questions that we will solve with the forthcoming estimators. How do we go from estimating the intrinsic dimension when there is only a single manifold in the data set to the setting where there's more than one manifold? Remember the colored data point with a different manifold. Well, from a statistical point of view, this is fairly simple because we go from a single Pareto distribution. This in red is the density of a Pareto distribution with parameter dk. We go from a single Pareto distribution to a mixture of k Pareto distribution, where k is the unknown number of manifolds. And then again, now that we have this mixture of Pareto distribution, we can estimate the intrinsic dimension either by maximum likelihood. So we take this mixture and we multiply, we take the product over the n data points, and we estimate by maximum likelihood the intrinsic dimension. Now the parameter is not just those k intrinsic dimensions, so there's one intrinsic dimension for each one of the k submanifold, but also those mixture probability, which in a sense are related to the number of points in each one of those manifold. Alternatively, we can take a Bayesian approach. We can put a prior on this vector of parameters, and we can compute the posterior distribution, which we know through Bayes' theorem being proportional to the prior times the likelihood, and we do the usual Bayesian approach, Bayesian machinery. And specifically, the prior on D that we use is a gamma prior. The prior on P, the vector of probabilities, this is typical when you have probabilities, you take your Dirichlet prior. And then, as it's often done when you have to estimate a mixture distribution, you introduce some indicator variables, Z, that tell you to which manifold each point belongs. And those are themselves parameters that you estimate. So without going into the details, let me get to some application. So the IRIS dataset is a dataset that certainly the statisticians that are listening know very well. In any case, we have 50 flowers of the iris type, and there are 3 different subtypes of iris: the versicolor, the setosa, and the virginica. And in this data set, we measure 4 quantities: the petal length and width, and the sepal length and width. So capital D is 4. In this case, so we have 50 flowers, so capital N is 50, and D, capital D, is 4. On each flower, we measure these 4 quantities. Now, in this example, we apply the mixture of Pareto distribution without telling that there are k equal to 3 subtype of flowers. So in a sense, we can also estimate k. This is the Bayesian nonparametric approach, and I will not go into the details. Either you take a Bayesian nonparametric approach and let k go to infinity, or you do a model comparison and you estimate the intrinsic dimension for k equal to 1, 2, 3, 4, and 5, and then you do a model comparison selecting which model best fits the data. But in any case, we don't tell the methodology that there are 3 submanifold, but the methodology itself identifies clearly that there are 3 submanifolds, which correspond to 3 clusters, and those 3 submanifolds are almost coincident with the flower species. So the first submanifold has intrinsic dimension 4, meaning that the setosa flower has very 4 degrees of freedom in varying how the flower looks like in those 4 measurements. On the other hand, the Virginica flower end up in the manifold of intrinsic dimension 3, most of them, not all of them. So the Virginica flower have only 3 degrees of freedom in varying those 4 measurements. And the versicolor flower has intrinsic dimension around 3.5. It's somewhere in between. We also tried this methodology on this simulated data set. Again, we don't tell the methodology that there are 5 manifolds, and we don't tell what is their intrinsic dimension. We let the methodology discover this altogether. And we clearly identify 5 manifolds, and the green line are the intrinsic dimension of those manifolds. So there's one that is very well identified, which is the one which has intrinsic dimension 9 and is very well separated from the others, and we pick it up very clearly. On the other hand, the black points, remember the black points have intrinsic dimension 1, but they're embedded, and it's very hard to identify their intrinsic dimension. But we identify a manifold that has intrinsic dimension 1, which where most of the first 1,000 data points end up, but some of them are wrongly assigned to the second manifold, which has intrinsic dimension 2, and some of the points in the second manifold are wrongly assigned to the first manifold. So those points are errors that we make. Still, we were very happy with the results because this is a very, very hard problem. We now move to addressing the issue of noise. So I told you here, this is again 3D points embedded in a one-dimensional manifold. Now, if we look at these data points too closely, zooming in too much, it really seems that their intrinsic dimension is 3. If we zoom out a little, but not enough, it seems that the intrinsic dimension of this point is 2. They seem to lie on a 2-dimensional cube. Only if we zoom out enough, we really see the one-dimensional structure of the data. So remember that with the grid estimator, we could let N2 grow. And so what we did is we look at the intrinsic dimension of this data, letting N2 grow, and or letting the number of neighbors that we consider for each data point increase. And we estimate the intrinsic dimension as K or N2 increases until we identify a plateau. And when we see this plateau, we report that value where the plateau is reached as the intrinsic dimension. So let me show you a different data set where the same problem arises. This is again noisy data constrained to live on this spiral. If we zoom it too much, so we have this lens, which is zooming it 300 times, the intrinsic dimension seems to be 2. If we zoom out enough, we really see that this data point lives on a one-dimensional spiral. So the intrinsic dimension is scale dependent. And again, we plot the intrinsic dimension. This is when we consider the 2 nearest neighbor estimator. It seems that the intrinsic dimension is 2. Then if we increase the number of neighbors to 32, this K and N2 are pretty much the same. The intrinsic dimension goes down and goes down again if we go to K 512. And only if we look at a sufficiently large neighborhood of each data point, we really see that we reach this plateau and we report the intrinsic dimension to be 1, which is the true intrinsic dimension of this data. But now the question is, sometimes you see this plateau, which is the intrinsic dimension that we report. On real world data set, sometimes you do not see this plateau. So the question is, what do we do if we do not see that the intrinsic dimension reaches a plateau? So these are data sets that are quite popular if you're in computer science, the NIST data set, the isomap are faces, and the isolate, those are recorded voices. So let me not go into the details, but in the isomap, we report the intrinsic dimension, we reach a plateau. Those lines are different estimators. Ours is the grid estimator, the blue line. Then we have the 2 nearest neighbor, and then those are other estimators that we compared available in the literature. So our estimator agrees with what is available in the literature in the isomap and the isolate dataset. But in the NIST dataset, you see there's no plateau. So what do we do here? Well, in this case, I have a slide also on computational efficiency, because we have to compute nearest neighbors. So this can be computationally challenging, but let me not go into these details. So I will go back to this question on how much we should zoom out at the end of my presentation. Let me show you some of the applications, real applications that we have looked at. So I told you that we looked at protein folding, fMRI, gene expression, some financial data, and COVID sport data, and so on. So let's focus on some of those applications. So this is a protein that goes from an unfolded to a folded state. You know, protein can change configuration, and the configuration of the protein is really important to determine the behavior of this protein, what the protein does. So here we have 32,000 configurations. So it is as if your data, capital N, is 32,000. And on each configuration of this protein that is going from a folded to an unfolded state, the other way around, from an unfolded to a folded state, we measure 32 dihedral angles. So capital D is 32. We use our methodology and we identify 4 sum manifolds. 3 of them have intrinsic dimension around 12 or 13, and 1 has a much higher intrinsic dimension, around 23. Now, when the protein is in its folded configuration, this corresponds with a higher intrinsic dimension. And so we can study the property of this protein also using this methodology. Another application is fMRI data. So in fMRI, you have voxels, and you take a certain number of scans of the brain, and can be resting state fMRI or fMRI when the person is doing something. So the data that we have analyzed, we have 30,000 voxels, and so capital N is 30,000 and capital D is 202 scans of this fMRI data. In this case, we clearly identify 2 manifolds, one of dimension around 16 and the other one of dimension around 32. And it turns out that the task relevant voxels, so remember this person is doing something, is a visual module task, and knowledge domain experts know which part of the brain is activated when a person is doing whatever she or he is doing. So it turns out that the task relevant voxel end up in the manifold with higher intrinsic dimension. So here in red, you see the high intrinsic dimensional voxel, in blue, the task relevant voxel, and in green, the intersection of the two. And because you see pretty much no blue, it means that all the task relevant voxel end up in the high dimensional manifold. So this again was interesting. Let me move to a financial application. Here we have 80,000 companies. Data is from some database. And on each company, we have 31 balance sheets indicators, some measure of the performance of the company from a financial point of view that tells us how healthy the company is. So we feed this data to our methodology. So in order to apply the methodology, you only need a matrix of data. So in this case, capital N is 8,000 and capital D is 31. Here we identify 4 sum manifold of increasing intrinsic dimension, 5, 6, 7, and 9. And then ex post, we go to the knowledge domain expert, colleagues that do finance, and we ask them, so can you tell us, can you see any common characteristic of the companies, for example, that end up in the manifold with small intrinsic dimension? Well, it turns out that the companies that end up in the manifold with the smallest intrinsic dimension, 5, are also the companies that have an S&P rating that is very low, D, C, or B minus. On the other hand, the companies that have a high intrinsic dimension, the ones that are have those balance sheets indicators that have more degrees of freedom to adjust and move, are those companies that have a high rating, A minus or A plus. And as the intrinsic dimension increases, the company become better rated from an S&P perspective. This is the Standard & Poor rating. And this, in a sense, makes sense. You can think of a company as like a body where we measure those blood, heart rate, and so on, pressure. Well, here on this company, we measure those 31 performance indicators. And when the company is doing well, those indicators have more degrees of freedom to be adjusted or to move around. On the other hand, when the company is doing bad, as a very low rating, those indicators are more constrained, have less degrees of freedom, smaller intrinsic dimension. Another interesting paper that was published in the Annals of Applied Statistics, and this was joint work with the colleagues from Queensland University of Technology, we analyzed data from NBA players' sensor tracking. So we capture 25 frames per second, and on each frame we have the coordinate X and Y of each player on the court, on the course, basketball, and we have the position of the ball. Here we have X, Y, and Z. So we analyzed data from season 2015-2016, and we were trying to understand what are the factors affecting the success of a play. And a play is an action with ball possession by a team. So often this data has been analyzed very much, but most of the summary statistics are static, and wanted to go from a static analysis to a dynamic analysis. Here I have a link. Okay, so here you see a play. The red is the ball, the green is the away team, and the blue is the home team. And you see how they move around and pass the ball. And for those of you who play basketball, you know that a team has some schemes, and if they manage to put forward this scheme, they usually are successful and go and score. But if the blocking team manages to break the scheme, then the attacking team has a problem. So here we see how the intrinsic dimension evolves as the time of the play goes on. And this structure of being low and then going up closer to the team scoring is something that we have seen in many of the play. I'll let you read the paper if you want to know more about this. Another application, which I think was very interesting, is to gene expression data. This is work with the Institute of Oncology Research of Università della Svizzera italiana. Here we have about 17,000 gene expression data on 69 tissues. So capital D is about 17,000, and we have only 69 tissues. The first 38 sample of tissues are cases. Those are patients that have a type of lymphoma. The last 31 are a control group. So again, we use our methodology without telling, the methodology that there are two groups and which statistical unit belongs to which group. And our methodology identifies very clearly two manifolds. And it turns out that the first 38 cases all end up in a manifold of the intrinsic dimension around 7 point something. All of them. And here you see some color coding, and I'll get back to this color coding, the blue and the black cases. And the last 31 controls, most of them end up in the manifold of higher intrinsic dimension, but it's not that high, it's 13. And remember that here capital D was 17,000. So a drastic dimensionality reduction. But some of the controls are wrongly assigned to the manifold of lower intrinsic dimension, the gray and the red ones. And I'll get back to this case at the end. So remember this issue, right? So we zoom out until we find a plateau. But the question is, what if we do not find a plateau? Can we find a better way of estimating this intrinsic dimension without relying on a visual, like a scree plot type of analysis? Well, yes, we can do better. And this is the last estimator, which is called the adaptive Bayesian intrinsic dimension estimator. We have the Bayesian and we have the frequent dispersion. I will not go through the code, but the idea of this estimator is that we start with the two nearest neighbor estimators. So we look at the two nearest neighbor estimators, so first and second nearest neighbor. And then for every point in the data set, we start increasing the value of k, the value of the neighborhood that we consider, until we have a stopping rule. And the stopping rule basically goes and tests whether the intensity of the underlying Poisson process is still constant. So we enlarge the dimension of the neighborhood that we look at until basically we meet the underlying assumption that on which we rely. And once the stopping rule is met, we return two things. One is the intrinsic dimension, but also for each point, we identify the largest nearest neighbor that we can look at, that we can consider. So this is an adaptive methodology and is specific for each point, because it might be the case that depending on the point that you are considering, so this is data set constrained to live on this spiral with noise. Like there's this point here where the largest value of k that you can consider is about 67, while this other point here, the largest value of k that you can consider is 212. But in any case, it's point specific. And it makes sense because there might be some part of your space, of your data, where you can, where this assumption of local homogeneity of the Poisson process is verified for larger balls or other points where it's the ball has to be smaller. So, so this is the adaptive estimator of the intrinsic dimension. So let me go back to this example of the gene expression. So the knowledge domain expert were very excited about our results. And they asked us, okay, good. So we started out with 17,000 genes. And now you tell us that there are really 7 or at most 13 dimensions that are relevant. First, they ask, okay, what are those genes? But really, it's not that there are 13 or 7 genes that are relevant. There are some function that takes as input these 17,000 genes and as as the domain, and the codomain is of dimension 7. So we cannot tell them what are the genes, but we can, in a sense, identify this function. It's a many-to-one function. So in order to, because they wanted to know, right, this map, because this is related, it has many interesting consequences if you're a genetist. So to do this, we took one of a very, very well-cited paper in Science, and this paper is about locally linear embedding. LLE is an unsupervised learning algorithm that computes a lower dimensional neighborhood preserving embedding. So it's a projection technique that tries to do exactly what we want to do, but takes as input this LLE methodology that has more than 20,000 citations. It requires 2 inputs. One, the dimension of the space where you want to project the data, and 2, k, the number of neighbors of each point that you have to consider in order to determine the data projection on the lower dimensional manifold. And these 2 things is exactly what our algorithm returns, because our methodology returns D and K. And more specifically, we do not only have D, but we have a Ki, a value of the largest neighbor that we should consider for every point. So given your data set that lives in capital D, now we have a methodology that finds the projected data for each point, its projection on lowercase d. So we start with X that lives in capital D, and we find the corresponding Y in lowercase d. So in a sense, for every patient, we have the projected genes in this lower dimensional space. So we went back to this gene expression data. Remember, 17,000 genes for 69 tissues. And now I tell you a little bit more about the color coding, but not much more. But the 38 patients are of two types of cell, ABC and GCG, activated B cell and germinal center cell. And these are the two colors for the patients, remember blue and cyan. And then the 38, those are healthy donors. They have 5 different colors, actually 6 different colors. And of those 6 colors, there are 2 types. Those are transitional plasma cell, TCP, and ACP, the advanced stage plasma cell. Those are being plasma cell represent the most advanced stage of B cell maturation. Now, you don't have to understand the details unless you're a genetist. And but okay, now this is the same data set that we have analyzed earlier, that I showed you earlier, now projected on a 2-dimensional manifold. So D star, the intrinsic dimension of the data, now we take it to be 2 using this adaptive methodology. And now you see the patients that are all grouped here, the blue and the black patients. These are all the controls, but the gray and red controls that are of this TCP and ACP type have their own cluster. So those are the ones that were wrongly assigned with the previous methodology. So I think I will stop here. There's another application on the tumor cell, the healthy cell, and this TCP and ACP cells, the plasma cells. I have another application on COVID-19 that I will skip. There's an application on the intrinsic dimension of convolutional neural network that we have analyzed. Actually, this is a paper of only the physicists. This is published in Advanced NIPS 2019. They studied the intrinsic dimension of data representation in a convolutional neural network. As you go deeper and deeper in the convolutional neural network. So across layer, they see that the intrinsic dimension first increases and then decreases. And the intrinsic dimension of the last hidden layer predicts very well, is very well related to the classification accuracy in the setting where this convolutional neural network is used to classify images. So just to give you an example, this is a convolutional neural network. There are different type of training, AlexNet, VCG, ImageNet, and we use 50 sample of about 20,000 images. And here is the intrinsic dimension as you go deeper and deeper in the layers of this convolutional neural network. And regardless of the type of convolutional neural network, you see that the intrinsic dimension first increases, then decreases. And at the last hidden layer, the intrinsic dimension is pretty much all the same and smaller than the intrinsic dimension of the input data. So if you want to use this methodology, I have good news for you. We both have an R package and a Python package. The Python package is maintained by the colleagues at CISA in Trieste. The R package is maintained by us. And to conclude, some references, pretty much all the methodological papers are in the Nature Scientific Reports, the open source journal. There's the Journal of Statistical Software for the intrinsic dimension paper. And there are some of the application papers that I mentioned throughout the presentation. Good. This is, yeah, thank you.
Okay, thank you so much, Professor.
Yeah, thanks for
wonderful talk. Thank you. Let's see if there is any question. Any question from the audience?
Yes, I have a question from a colleague who is sitting here in...
Okay.
Yeah, please do. So let me stop sharing. Here we are. So thanks for all the students or colleagues connecting online. Yes.
So I have a semi-infinite number of questions, but I start by a few ones. So
actually, let me introduce you. This is a colleague, Professor Caccia. Maybe you can introduce me.
Hi, Professor.
I'm a physicist. Actually, I'm an experimental physicist. So my first question is related to who I am and what I do. because when you are estimating D, this will never make a physicist happy because you're not attaching an error.
I don't know. Of course, we have an error.
You have an error.
Of course. I'm a statistician. I always have
Okay.
errors with my estimators. So we have a point estimator with a confidence interval, and we also can do hypothesis testing.
Okay.
So if I can tell you if the intrinsic dimension of a data set is larger or smaller, I can give you both a p-value or I can be Bayesian and do the test.
The
So
I
do,
presume,
No, no.
for instance,
No, no,
is
I
that
do have
you're
an error.
changing the noise, then the confidence level changes.
but the noise is in the data.
Yeah,
Okay, so
I
there's
mean,
a noise level.
if-
Yeah, of course,
If the
yes,
level
yes.
of the noise in the data decreases or
Yes,
is
absolutely.
inflated, then I believe the
Yeah,
confidence level
absolutely,
changes.
absolutely. Yes. So
Okay.
first, yes, thank
Follow the data. I do not